Welcome to the introduction of one of our new systems, the skeletal system. Here you'll find that our discussion in lecture is going to deviate from that in lab. Lab is going to mostly deal with bone identification, bone markings or bone features. Every little crack and crevice on a bone has a distinct name, and there will be plenty of them that you will have to be able to identify visually. In lecture, we're going to stick more with the physiology of the bone. We'll spend much more time looking at bony tissue, how that tissue grows, how it develops, how it elongates, how it transitions into mature bony tissue. Before we get into that, however, we kind of have to refresh a little bit and update some information that we'd already mentioned when it comes to connective tissue, because our skeletal tissues are primarily going to be connective. So we have to know where we distinctly have left off with connective tissue and where we're going with it now. You'll have to know the processes involved in growth of cartilage and bone. They share some terms, but the process is obviously different. We'll look at the regional structure of a bone, particularly of a long bone. Within that bone, we'll see we have two different types, compact and spongy bone. We'll look at the processes of ossification and distinguish it from some other more simplistic processes. We'll look at long bone growth at the growth plate, as well as the factors involved with growth, healing, and repair. So as we go through our introductory skeletal system topic, we're going to be looking at the tissues involved with bone, cartilage, ligaments, and any other associated connective tissues. Now, we used connective tissue previously in a very vague tissue context when we talked about epithelial tissue, connective tissue, muscle, and nervous. You'll see in a moment, once we look at our subclassifications, that was a classification of connective tissue all in of its own. And what we're going to be moving into today is more distinct connective tissues from an organ context. Back when we did tissues, we talked about connective tissue function, and we gave it a varied array of different functions. We said, generally speaking, we saw connective tissue being supportive and protective. And that's not going to change here. We'll even see those roles flow over into cartilage and bone in particular. But we also saw connective tissues be involved with storage, transport, immune function. And some of those functions had very tissue-like roles that we won't see carry over into cartilage and bone. Cartilage and bone will take on their own very particular functions. Here's an arrangement of where we've come from and where we're going next. You might see some terms on the diagram here that look familiar. Areolar connective tissue, adipose connective tissue, dense regular and dense irregular. We used this term back then, but to really highlight it now, all of those were connective tissue proper. Really non-specific from an organ standpoint. We generally saw connective tissue proper associated with other organs outside of the skeletal system. Not that they can't be involved in the skeletal system as well, but we looked at connective tissue proper purely from a tissue standpoint and as them being a component of other organs. Our supporting connective tissues are where we're going to spend our time now. Cartilage and bone. And as you can picture with these, especially bone, we have a femur sitting in front of us, for example. Now our connective tissue discussion kind of flows into more of an organ level. And we see it on the diagram, so we may as well mention it. Blood, also a connective tissue. But of course, you won't be getting into any blood material until you get into cardio as part of your AMP2 discussion. We said that every connective tissue has a fibrous component. Actually, we said that as a cellular component, a ground substance component, and a fibrous component. But we didn't really get into the different types of fibers. But if we're going to understand the different types of cartilage and bone, that's kind of where we have to go now. The first type is a collagen fiber. Long, unbranched, very strong, rather flexible, however, very resistant to stretching. And if you do manage to stretch it, you're probably damaging the collagen fiber. So when you relieve the force from it, it won't stretch, it won't snap back. It doesn't have any elastic quality. And that's why you see, as collagen fibers break down in structures like the skin, typically the end result is going to be a wrinkling. Elastic fibers, as mentioned, are elastic. Now, I want to make sure we're aware of what elastic means. Elastic does not mean the ability to stretch. Stretching means elongation. Elasticity is when we remove the forces that elongated a tissue. Elasticity would be that for that tissue to spring back to its original length. That is elastic. And that's what we see with these particular fibers. They're thinner, they're not as strong, but they do have that elastic property. Reticular fibers are going to be the thinnest and weakest of them all. They provide some structural support, but they're never really depended on to provide structural support for an organ that undergoes any significant wear and tear. More so like they help maintain the shape of an organ rather than resist any force. Think about the network of reticular fibers that may work their way through a liver, a spleen, or a lymph node. Cartilage is a semi-rigid connective tissue. So we're talking about that ground substance component remaining a very flexible structure. Now we have two different types of cells that we'll typically see in cartilage, and we need to break down the nomenclature. First of all, when you see the prefix chondro, that means cartilage. So if you're given a question that asks which type of cell do we see, whenever, if you know to refer to a cartilage, you want to look for one that has the prefix that's relevant to it. The rest of the term, in a lot of cases, will refer to maturity of a cell. Chondroblast being an immature cell, cyte would refer to a more mature cell. However, in this case, we want to get away from that distinct definition. For us, a blast is going to be a cell that lays down new matrix. But remember, we're dealing with a semi-rigid structure. As it lays down new matrix, it's running out of space, painting itself into a corner, so to speak. At the point when it no longer has any room to lay down new matrix, then we call that same cell a chondrocyte. And the space in which it resides is the lacunae. From that point on, the chondrocyte is not responsible for laying down new matrix. They're simply responsible for maintaining the matrix immediately around their lacunae. So as I described that, we had our chondroblast turn into a chondrocyte. But also perceive that if we got some cartilaginous damage, and now all of a sudden this chondrocyte had more room to lay down new matrix, it could. And if it's laying down new matrix, now we simply call it a chondroblast again. So the terms are really more descriptors to what the cell is doing in the moment. If that cartilage cell is laying down new matrix, we call it a chondroblast. If it's not, we call it a chondrocyte. And depending on what that tissue is going through, we could bounce back and forth, name-wise, between each of these. We have three different types of cartilage we're going to need to know: hyaline, fibro, and elastic. Now pay attention to the pictures here, because we're going to be looking at very distinct things that will even help you in lab and identify them on a slide. First off, is I want you to look at the hyaline cartilage, the one at the top. Very shiny, glistening, almost like a gristly appearance. Very little fibrous matter apparent. I mean, it has a fibrous structure, but on a slide, the way it's stained, it won't really look very fibrous. You will see these chondrocytes in their lacunae, suspended in a very clean, sterile-looking ground substance. Very flexible, and just as the appearance would suggest, very smooth. So we see hyaline cartilage make up the joints where the ribs meet the sternum, the tracheal rings that support the windpipe. But based on its appearance, the one I would suggest you always try and remember is the articular surfaces. Wherever one bone meets another bone to form a joint, those pieces of bone will have a nice layer of hyaline cartilage, forming a nice, smooth, friction-free or friction-reduced surface for those joints to glide over. And when you look at that tissue under a microscope, it simply looks smooth. Helps you adapt structure to function. Now, when we switch gears and look at the fibrocartilage, you see a lot of fibers. You see almost all fibers. Very resilient tissue. And the density of the fibers should allude to that. And look where we find it. Pubic symphysis, meniscus of the knee, intervertebral discs of the spine. Now, the pubic symphysis, not so much, but could you think of joints that get more physical wear and tear than the knee and the lower spine? Very resilient to wear and tear. That's where we want them. Physical appearance on a slide, very dense with fibers, distinguishing it from the hyaline cartilage that we just mentioned. Now, last up is our elastic cartilage. Now, you'll tend to see another very fibrous tissue, but not nearly as densely populated as our fibrocartilage. And even the fibrocartilage, the fibers will be running very much in a parallel arrangement, not so much with elastic. We see elastic in two very distinct places, the epiglottis of the larynx and the external ear. Now, we haven't gotten to the epiglottis yet. You will when you do respiratory. That is a spoon-shaped piece of cartilage that every time you swallow, it folds down and covers up the opening to the windpipe. So whatever you're eating is not going down into the lungs. Think about how that elastic quality is important. The thousands and thousands and thousands of times you chew food and swallow, chew food and swallow. We want that epiglottis to keep snapping back to its original shape. Now, while the external ear, its composition of elastic cartilage might not seem quite so useful, but it can give you an impression of what that cartilage can do when you fold the external ear down and you let it go. Fold it down, let it go. Keep snapping back to its original length. Cartilage will be very supportive. We already mentioned its role in articulations. And later on, you'll see us get into it being a precursor for bone. One of the types of bone development we'll talk about involves using a template made of hyaline cartilage that then we change into bony tissue. We see two means in which cartilage can grow. We have interstitial growth and we have appositional growth. Now, the process is not going to be that different. Where I want you to focus on is where the growth occurs. In interstitial growth, the most active cells are always going to be the ones deep in the mass of cartilage. They're the ones undergoing mitosis, laying down new matrix. So if you have a mass of cartilage in your hands that have been growing for a while, exclusively by interstitial growth, The tissue on the outside of that mass is the oldest, with the newest tissue always forming deep. Appositional growth is the opposite. The most active cells are on the surface of that tissue, under a membrane called a perichondrium. Peri meaning around. Now, in this case, you get a mass of tissue. The newest tissue is on the outside of that mass, with the oldest tissue being deep. Now, the drawings I have up here are supposed to be of both of those processes, but I'm not going to use this diagram for it. We'll be coming back to this, actually, once we look at long bone growth. We'll be referencing this chart. Because look at what generally happens. We even see this in both types. We get a chondrocyte in a lacunae. It undergoes mitosis and splits. So now we got two cells in the same void. We wall off that lacunae to form two separate ones, and now those lacunae start to drift apart. And that's how we'll see cells undergo mitosis and spread through this tissue, increasing tissue size as it goes. Now we move into bone tissue. And we need to get a couple of clarification things out of the way right away. First off, bone is very sophisticated in its structure. It has a very particular arrangement. You'll see that when we talk about bone structure in lecture. You'll see it on microscope slides in the lab. There seems to be this thought amongst a lot of people that bone is this inorganic scaffolding that happens to be in the body. But bone is very much a living tissue with a very robust blood supply. So terms such as calcification, we want to get away from thinking that they even really apply. We'll be mentioning calcification, but bone is not simply calcified. We throw three terms at you here now: mineralization, calcification, and ossification. And kind of moving up into levels of sophistication as we go. Mineralization is when we simply deposit any number of inorganic minerals into a soft tissue. Calcification is a type of mineralization, but this time specifically for calcium. Ossification is the particular arrangement of ground substance we need to make bony tissue. Any soft tissue can become calcified. If we did an x-ray of everybody in the class, we would see some people with bone spurs on their heels and on their spine. The x-ray would look like bone growth and like a bird's beak appearance. That's not bone. That's simply calcium deposits lodged into a tendon or a piece of cartilage that now makes it look more rigid and appear as bone on an x-ray. But that deposition of calcium into those tissues has no regular arrangement, has no particular structure, and you would see tendons become calcified, carotid arteries become calcified, you get calcium deposits in muscle, any number of things. Ossification is very specific in its arrangement, and you'll see how specific once we look at the structure of bone. Functionally, we still see support and protection hold over. Right? It really is a hallmark of any connective tissue in some way, shape, or form. Going to be responsible for movement. Now, not the contractile portion, obviously. That's muscular. But if you want to undergo locomotion, you kind of get dependent on both, right? With the bone providing the rigid framework, the muscle providing the contractile tissue that anchors onto it, and together we get effective and efficient locomotion. We see hemopoiesis as a role. Now, you won't get to this distinctly till A&P2, but hemopoiesis is blood cell production. We see red blood cells produced in the bones of the humerus, the femur, the pelvis, the vertebra, the sternum, and the ribs. We see some white blood cell production in the bone marrow of the long bones. We see some fat deposited into the yellow bone marrow, but I hesitate to call it an energy reserve, so to speak. So it's not really a place we get the deposition and withdrawal of fatty tissue in order to save energy for a rainy day, so to speak. But we do use the skeletal system for the storage of various minerals, and the most well-known one there is going to be calcium. We will talk about calcium balance with two particular hormones, calcitonin and parathyroid hormone. When you look up the definitions of those hormones, calcitonin is going to reduce blood calcium. Parathyroid hormone is going to raise blood calcium. But the question you should be asking after that from a skeletal standpoint is, where is it getting it from, or where is it shoving it to? If calcitonin's job is to reduce blood calcium, one of the ways it can do that is by taking that blood calcium and depositing it into the skeletal tissues. Parathyroid hormone wants to raise blood calcium back up at times when it's low. In order to do that, it can tap into the stores of the bone, release some of that calcium, and when that calcium gets released from the skeletal tissues, it spills over into the bone, into the blood, raising blood levels back up. Now, we also have a distinct set of cells in bone, as you can imagine. First is the osteoprogenitor cell, an immature stem cell for bone. Osteoprogenitor cells will always mature into osteoblasts. Now, to get our nomenclature out of the way again, osteo means bone. You've heard of terms like osteoporosis before. Blast takes on the same meaning we saw with cartilage. Lays down new matrix, lays down new ground substance. It's involved in bone growth. But just like we saw with cartilage, and even to a greater extent with bone, it can only lay down so much tissue before it paints itself into a corner. Then, just like with cartilage, we call it an osteocyte, and it resides in a lacunae. Also, just like cartilage, we can jump back and forth between these two names, depending on what the bone cell happens to be doing. Again, these endings are based on what the cell is doing, not simply based on the age of the cell. Now, if you'll notice, we have one more cell left on the list. I even distinctly left a space here. The osteoclast. Another bone cell, but in no way, shape, or form familially related to the first three. The first three does kind of move in this maturation sequence, but the osteoclast is a different animal. It's a modified white blood cell that happens to also reside in the bone. Now, I want you to think of osteoclast as the direct antagonist to the osteoblast. Osteoblast lays down new bone involved in bone growth. Osteoclast dissolves bone and breaks it down. So both of these cells will obviously be involved with that calcium regulation that I mentioned earlier. Calcitonin wants to reduce blood calcium, so it's going to activate the osteoblasts to deposit calcium into the blood. Parathyroid hormone wants to raise blood calcium, so it activates the osteoclast to dissolve that bone, releasing its minerals out into the circulatory system. Now, even though we stressed that bone wasn't a simple inorganic framework, it obviously is composed of some inorganic matter to make up the ground substance, particularly a high degree of phosphates and calciums. But I really want to make sure that we are well aware that ossification is the process of laying down a very particular arrangement of this ground substance. We have two different types of bone as far as their appearance goes. Compact bone, spongy bone. Compact bone is very dense, resistant to physical impact and wear and tear. Any bones that undergo a lot of stresses and strains are going to have a high composition of compact bone. Spongy bone, not meant to imply that this bone is soft. It is not soft, not bath sponge. Think more along the lines of a pumice stone or a lava rock. It's very porous. It's hard, it's rigid, but it's porous. And as a result, it's simply not as dense. That porous structure we call trabeculae. Now, in a flat bone, we'll see them arranged in a very particular manner. We have compact bone lining the external and internal aspects of that bone. And in almost like a sandwich kind of arrangement, the spongy bone kind of makes up the meat of the sandwich. So we have compact bone anywhere where that bone would get abuse, a bump on the head, for example. That's going to be absorbed by the dense compact bone. When we look at the compact bone, there's a very regular arrangement, actually divided up into subunits called the osteons or the Haversian systems. These are long cylindrical subunits that typically will run parallel with the long bone. So if we picture a long bone, picture your femur. This is not a femur, but picture your femur. Every time you jump up and down, the stresses and strain along the cylinder of that bone has these long cylinder-shaped osteons running parallel with it, absorbing that force. Now, we have a laundry list of terms we need to know with compact bone, and I've subdivided them up to make them easier. The ones on this slide all have to do with the osteon. These are osteon terms. So if we skip to the diagram without me reading those out, we'll go through them while we're looking at the osteon. Here we see it from above, this circular arrangement. But also remember, it's a cylinder. So this would be like looking at a soup can from the top, but recognizing it has some depth to it, and it runs parallel with the long bone. The first structure is the central canal. It's an opening right in the middle of that osteon. Remember, bone is very hard and rigid, but it's also very vascular. So we're going to need a network of tunnels to allow blood vessels and nerves to penetrate that tissue. The central canal is one of those. Surrounding the central canal are these complete circular rings that surround the osteon and the central canal in layers. These are concentric layers. Concentric means radiating out from a point in a layered fashion. These are concentric rings around the central canal. They're called concentric lamellae. Lamellae referring to the layered arrangement. In between the lamellae, we see lacunae. If you remember from what we just talked about, each lacunae now is going to have an osteocyte. Those cells inside the lacunae are going to need nutrients. They're going to be producing waste products. So we have this network of very fine canals going into and out of the lacunae called canaliculi. So between the central canals and the canaliculi, we get a way of nourishing the cells that are in this very dense tissue. Spongy bone has no osteons. However, when we look at the trabeculae of the spongy bone, we see it's still oriented in rings, but no central canal. Without the central canal, we can't call these concentric lamellae. So we need a new term. In spongy bone, these are parallel lamellae. So concentric lamellae associated with the osteon, parallel lamellae associated with the spongy bone. We're getting there. We've got more terms. If we look at the remainder of the bone, you will see more layered structures, particularly here. These are rings that go around the entire diameter of the shaft of the long bone. They run the circumference of that diameter. Therefore, they're going to be called circumferential lamellae. And then finally, filling in the spaces here. Because obviously, if an osteon is a cylinder shape, cylinders don't like to pack together tightly. So we fill in the spaces in between the cylinders with more layered structures. We call those interstitial lamellae. Now, to round out one more term, we had our central canals running lengthwise with the long bone. Running perpendicular to them, we have perforating canals. So the perforating canals link up each central canal, so we get a nice tunnel-like network for blood vessels, nerves to penetrate through that tissue. We don't have the need for that tunnel system with spongy bone, remember. Inherently in spongy bone, we have a tunnel system. It's a very porous tissue to begin with. So we will just run blood vessels through the trabeculae and kind of interweave blood vessels through that spongy bone without needing a particular formed tunnel network. Now, I said we would get to ossification and bone tissue development. Well, here we are. We've got two different patterns of ossification, but they both wind up producing particular ossified bone, as we had in those last slides. First off, intramembranous. Intramembranous develops directly from a primitive tissue called the mesenchyme. And we only see intramembranous ossification in a couple places in the body. The flat bones of the skull, some of the facial bones, the mandible, and in the weirdest pattern ever, only the middle portion of the clavicle, the central third of each collarbone, undergoes intramembranous ossification. So we take our mesenchyme and directly convert it into osseous tissue. The second type is endochondral. That's what we alluded to earlier when we said we take this cartilaginous template. We take a piece of hyaline cartilage that roughly looks like a bone, and we run it through a process where we convert that cartilage tissue into true ossified tissue. Now, this is going to be all the rest of the bones, including the proximal and distal ends of the clavicle. Now, when we go through these, and even when let's throw fracture healing in there for a moment too, you are going to see a generalized pattern that I want you to keep in mind. It'll help you remember the specifics of each process. We see the same pattern occur with both of these types of ossification: growth at the growth plate of the bones and fracture healing. We see us go from a cartilaginous tissue, or a soft tissue in general. Then we will calcify that soft tissue. Then we will take that calcified soft tissue and turn it into ossified tissue. That is the general chronological sequence of events we're going to go through. So let's look at that with the intramembranous. The first thing we get is the formation of an ossification center. We get osteoblasts kind of huddle up, group together. They're going to start secreting substances into their immediate surrounding matrix and form this osteoid. So we have this osteoid with these osteoblasts suspended in it, making up this entire ossification center. Now, as promised, we take that osteoid, that soft tissue that in the slide previous would have been a semi-rigid structure like a cartilage, and we calcify it. We simply deposit minerals into the osteoid. Now you'll start to see those osteoblasts be named osteocytes. You got this calcified osteoid right immediately around it. But on the periphery, you're still seeing osteoblastic activity and uncalcified osteoid. So we're taking this ossification center now, and you're seeing ossification spread from that point as we calcify new osteoid as we go. Now, the first osteoids that were developed are going to start undergoing the ossification process. First, we turn it into what's called woven bone. Now, already, if you picture something that's woven, picture a basket that's woven. There's going to be a layered context to that, right? Remember, when we dealt with spongy bone and compact bone, we had a lot of lamellar structure. This woven bone is a very simplistic version of that so far. We take that woven bone and we make it a more sophisticated. Now, all of a sudden, we can call it lamellar bone. And that's where we're going to start getting the distinction between these concentric lamellae, circumferential lamellae in the compact bone, and the parallel lamellae of spongy bone. Soft tissue, we calcify that soft tissue, we ossify that tissue. Then we have bone. We see the same general process when it comes to endochondral ossification. Let's look at it a little bit more specifically. Remember, we start with a piece of hyaline cartilage. It even looks roughly, maybe not bone shape, but dog biscuit shape or something that's meant to look like a bone, right? The first thing we start to get is we'll call it periosteal bone collar surrounding the outside of that bone. From there, we see the term ossification center pop up again. We get an ossification center form roughly in the middle of that bone. And just like we saw with the intramembranous, we see a spread. But this time we see a spread lengthwise. We're going to see that ossification process migrate towards the proximal and distal end of that long bone. While that's occurring, we get other bone centers occur, secondary ossification centers. So we're ossifying the polar ends of that bone, we're ossifying the middle of that bone, and we're working towards those ends. Eventually, we're going to be left with this piece of cartilage remaining that's got bone on both sides. That last bit of hyaline cartilage is our epiphyseal plate, or our growth plate. That, moving forward, is the only place where we can get activity to make the bone grow longer. Once that epiphyseal plate fuses, your long bones will not get any longer. You're as tall as you're going to get. There's no way to reactivate those growth plates. But you can still see how we went from soft tissue, in this case a hyaline cartilage, we calcified that soft tissue, then we ossified that calcified tissue. When we look at the epiphyseal plate, we're still going to see that same general process, believe it or not. The epiphyseal plate is going to be divided up into 5 very distinct zones. Now, the zone of ossification, zone 5, is always going to be closest to the shaft of that long bone. The zone of resting cartilage, zone 1, is always going to be close to the ends of the bone. So picture that if we look at this long bone, we have 2 different epiphyseal plates here. Here, zone 5 is going to be closest to this side, and zone 1 is going to be in that epiphyseal plate, but closest to that side, or end. On the lower part of the screen, those have to be inverted now, because what we're showing you there on the slide with the layers, this is not a superior-inferior labeling. If we are talking about your humerus in your upper arm, this could be the order of the zones of the epiphyseal plate as we go from proximal to distal. However, if we were talking about that same bone, but the epiphyseal plate down close to the elbow, this would be the zone closest to the elbow, and now we would be moving from distal to proximal. So rather than memorize these terms, I want you to understand the terms, because the name here tells you what's going on. It's very descriptive. First of all, zone of resting cartilage. Now remember, this started off as hyaline cartilage. The resting term implies that nothing's happened yet, and rightfully so. The zone of resting cartilage will still look like hyaline cartilage on a slide. No distinct difference. Zone 2, proliferating cartilage. The term to proliferate. When we're talking about that from a cellular standpoint, we're talking about cells that are dividing rapidly. Now, because of the rapid mitosis that we get here, we get the cells and the lacunae arranged like stacks of coins. Now, that's what I wanted to get into. I said earlier we would look at a slide. We'd come back to this one. Remember in cartilage, when these 2 cells divided, then they walled off their lacunae, and then we had time for those lacunae to drift apart. If these cells are dividing extremely rapidly, the amount of drift we get here is going to be minimal. So what you wind up getting in this zone of proliferation is the lacunae stacking, because as soon as that cell divides, we're not having enough time for the lacunae to drift apart. And the lacunae that are formed remain huddled close together in this coin stack looking formation. Next up is the zone of hypertrophic cartilage. Proliferation meant to divide rapidly. Hypertrophic means to grow in size. Now these same cells that were undergoing this rapid mitosis, now they've got a moment to slow down their replication. Can't have a big cell that's replicating quickly, right? By the time it gets to its interphase, it's dividing again. But now that mitosis has slowed down, giving these cells a chance to get larger. And as you can imagine now, with that increased time, the lacunae that were forming this coin stack appearance, they're going to lose that appearance now. Those lacunae are going to have more time to drift apart. Zone of calcified cartilage. Remember I said the overall theme: soft tissue, calcified tissue, ossified tissue. Now that cartilage is going to become calcified, and we take that calcified tissue just like we did in bone growth to begin with, and we change it into true ossified tissue. When we look at that on a slide, we should see resting cartilage. Looks just like any other hyaline cartilage. Here is the coin stacking we were referring to. See how we're getting these proliferating cells multiplying rapidly so that the lacunae now are stacking on top of one another. But once the cellular division slows down, now we see a distinct change in the size of the cell, and we start to see a loss of that very well-arranged stack-like structure. We calcify the tissue, and then we ossify the tissue. Now, as we go through lengthwise growth of a bone, realize the thickness of the epiphyseal plate. If we were to measure the epiphyseal plate, and let's say, just for argument's sake, it's a 1/2 centimeter thick. As the bone grows, and as you go from someone who's 3 feet tall to someone who's 5 feet tall, the thickness of that epiphyseal plate stays relatively constant. It's not like the taller you get, the thicker the epiphyseal plate gets. The processes that are occurring in each of those layers work like a road crew. And what they're leaving behind is ossified tissue, the same way a road crew would leave behind asphalt. We got 5 layers, each with a distinct role in that ossification. If we had 5 teams of people working and laying down new road, the first team could be the bulldozer guys who are going out into the farmland. 500 feet behind them could be the guys in the grading tractors smoothing the surface out. 500 feet behind them could be the guys laying down this crushed stone and smoothing it out. 500 feet behind that group, you've got the people laying down the tarmac, the asphalt. And the last group coming through is painting lines on the road. 5 groups of people with 500 feet in between each group. We got 2,500 feet of road work. But as we move across acres and acres and acres of farmland, the first group don't get that much ahead of the last group. They march across the farmland, keeping the same distance between them. And all they've done is left behind longer and longer and longer stretches of road. This epiphyseal plate leaves behind longer and longer and longer stretches of long bone shaft, making that long bone taller. What we just described there was interstitial growth of a long bone. So this is handy when we get to give you a term after we've already talked about the process. Interstitial growth of bone refers to the bone's change in length. Now, we used the term interstitial growth earlier, right, as opposed in the context of cartilage. This is different. Cartilaginous interstitial growth was growth deep in the mass of tissue, giving us a bigger piece of cartilage. In bone, that term applies specifically to elongation. For appositional growth, it involves growth in thickness and diameter. Now you see the term periosteum. We haven't even gotten to that yet. Periosteum is a membrane that surrounds the outside of the bone. And we have another membrane surrounding the inside of the bone called the endosteum. Now again, we're putting the cart before the horse here just a little bit, but it's relevant. Those 2 membranes take on 2 different roles. In order for bone to get bigger, we have to have osteoblastic activity. That should make sense. Remember, osteoblasts lay down new bone. So if we want that bone to be thicker, we have to lay down more osteoblastic activity. But in order for a bone to get bigger, we also have a certain degree of osteoclastic activity. And that's the part of this that people forget. We have osteoblasts being active around the periphery of the bone, where that periosteum is. But while that bone's getting bigger, the cavity inside the bone is also getting larger. So we need osteoclasts in the endosteal membrane, widening out that medullary cavity. We have any number of hormones, vitamins, and processes that can change bone remodeling and growth. It's not going to be a big topic that we'll talk about here, but we're going to introduce it. We already mentioned some hormonal activity with calcitonin versus the thyroid versus the parathyroid hormones for calcium regulation in the blood. Some others that we could talk about: growth hormone. Shouldn't be any mystery as to what growth hormone does to bone, right? Stimulates growth. If you're young and your epiphyseal plates are still active, it'll make you a lot taller. If you're older and those epiphyseal plates have closed, now it'll make the bone thicker and denser. For the vitamins, I want to stress that we don't necessarily use vitamins to trigger osteoblastic activity. It's not like if you're young and you want to get taller, you just start eating vitamin A. It doesn't work like that. More so, if we want active osteoblasts, then we have to have vitamin A available. If we want to lay down collagen, we have to have vitamin C available. But it's not like pumping yourself full of these vitamins will cause these processes to occur at a greater rate than they would have naturally. Last up is the effects of exercise. Or at least last up as far as bone remodeling. Bone is a very active tissue, and it will remodel and reorganize itself based on where the stressors are. It's called Wolff's Law. Use it or lose it, basically. If you are putting your bones under significant physical stress and strain, they will become thicker and stronger. If you lead a more sedentary life, they will get weaker and thinner. We see bone demineralization in people who are quadriplegics. They can no longer walk. They can't put stress on their leg bones anymore, so they become naturally weaker. It's a significant problem to people who have spent quite a bit of time in zero gravity, astronauts, space center inhabitants. Long periods of time in zero gravity, you're removing stress from the bone. So being in that situation, it's important for them to do particular exercises to help maintain bone density. When we look at fracture repair, I kind of want you in the back of your mind still thinking about bone development, right? Soft tissue, calcified tissue, ossified tissue, because we kind of run through a very similar process. Remember, bone is very vascular. When you fracture bone, think about all those blood vessels in the perforating canals, in the central canals. They're going to be sheared off. So you will get a hematoma formation. And you will go through the process of blood clotting and vascular spasm because it's a vascular injury. That very soft tissue now is going to be converted into a cartilaginous callus. Now, you see the term fibrocartilaginous callus. This is not to imply that this is fibrocartilage. Sometimes the terms here can be your own worst enemy. This is fibrocartilaginous, not fibrocartilage. It's a fibrous, cartilaginous substance. Soft callus. We're going to take that soft callus and we're going to calcify it. Then we're going to remodel that callus into a bony callus. Now at this point, that's ossified tissue. And through ongoing physical stress on that bone after the fact, that bone will remodel until hopefully it's back into the same arrangement that it was when it fractured or before it fractured. With respect to classification of fractures, there's any number of ways you can describe it. You can describe the angle that the fracture goes through, transverse, oblique, in a twisting manner. Spiral kind of implies the mode of injury. You can describe what it does to the surrounding tissue sometimes, like a compound fracture is when a piece of the bone penetrates the skin. Open fracture. A comminuted fracture is when you get several bone pieces. And depending on where the bone is, you may even have specific terms used to refer to comminuted fractures. Like a comminuted fracture of the vertebral body can be referred to as a burst fracture. If you're really well known, you can start describing patterns of fractures and get your name associated with it. Colles, a fracture of the wrist often seen with what they call a Fuchs injury. Fall on outstretched arm or fall on outstretched hand. You slip on the ice, rather than land directly on your tail, you stick your hand back to catch yourself, and of course you get a fractured radius where the distal portion is posteriorly dislodged. That's a Colles. A Pott's fracture is a fracture of the fibula and the tibia on the same leg with a disruption of the joint at the talotibial area. Now, the one I want to focus on, this is the one I'll ask you questions on, I'll just be blunt about it, the green stick fracture. This is a fracture in an immature bone where it doesn't snap like a dry twig. It'll break like a green stick pulled off of a healthy tree. If you take a healthy branch off of a tree and try and crack it, it bends more than it breaks. And what you'll get as you bend it, you will still see intact branch along its concave side, but then you will see a split form in the convex side. That's a green stick fracture. Now, there's any number of different types. An avulsion fracture is when you pull the tendon and it pulls away from the bone. A stress fracture or a microfracture or a hairline fracture is a fracture you get with repetitive trauma over a long period of time. You can have pathologic fractures that might be secondary to tumor or osteoporosis. So there's any number of names. We're not going to make that a big part of our class. I'll be asking you about the green stick fracture. Now, osteoporosis, we can try and take some of the things that we discussed earlier and kind of put them in this context. We won't look at aging as a factor of every system we do. And even here, it's not even so much aging as much as it is neglect over a period of time in a lot of cases. This is when the bone becomes demineralized and weaker. So if we look at things that could lead to it, we already talked about some hormones, right? Loss of hormone activity. In females in particular, estrogen. Post-menopause, estrogen drops. Estrogen is a hormone that supports bone deposition, so the bones get weaker. Lack of exercise. Someone who hasn't stressed their bones much. Now they're more elderly. Now it's going to be more of a problem. It could be dietary. Whether or not eating adequate resources or they're not absorbing it. Again, the elderly might not be absorbing things like calcium and vitamin D as efficiently as they were when they were younger. But regardless, a decrease in that bone density often increases the chance of fracture. And one you hear about all the time is the hip. You hear people say they fell and broke their hip, when in reality, the sequence of events that occurred there was most likely they stood up, their hip broke, then they fell. So a lot of terminology in this section. Some stuff to understand, but a lot of structural terminology. Make sure you don't get the terms confused between cartilage and bone when we're talking about the same word. Make sure you know the structure of the compact bone, both in diagram and for verbal questions. You should know the activities of the epiphyseal plate. The 5 layers, what happens at each layer, the order of the layers, and the process of growth for both bone and cartilage, as well as the 2 different types of ossification. As usual, if you have any questions, please reach out, and we'll move into gross anatomy of a long bone next. Thank you.